back to my channel Biomixup. In this today's video, we are going to discuss about the very important enzyme of DNA replication, that is the DNA polymerase. In the last video, we have already discussed about the DNA replication. What are the enzymes used during this DNA replication? DNA polymerase is also one of the enzyme. It is the main enzyme for the synthesis of the DNA. So DNA polymerase, they are of different types, like it can be DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, 4 and 5. There are different type of DNA polymerase. During the process of DNA replication, mainly 2 to 3 enzymes are responsible, like DNA polymerase 1, 2 and 3. Basically, DNA polymerase 3 are used during the process of elongation. This DNA polymerase 4 and 5, they are used during the repair mechanism. When there is any incoming nucleotide which is having mismatch, they are being repaired by DNA polymerase 4 and DNA polymerase 5. So let us see how the structure of DNA polymerase. This DNA polymerase, it is like a palm-like structure. We can see here in this diagram, this green color diagram, it is like a palm. Here we can see the finger-like structure. This is a finger-like structure and this is a thumb. Okay. What happens when the DNA is replicating? What it does? It have to convert itself into a single-stranded DNA. Okay, so this work is being done by DNA helicase. So during this time, this is the, we can see that this is the parent DNA. This is the parent template and this green one is the daughter strand. This green one is the daughter strand. During the process of replication, this DNA, when the DNA polymerase enters into this DNA is showing little bit of bending like structure. We can see here, this is a bending like structure. This is the parent DNA. This is the parent DNA which bends, and the upcoming nucleotide is being added in this 3' OH group. Okay, so let us see what are this DNA polymerase. What are the function of this DNA polymerases? Like we will discuss here about the DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. DNA polymerase 1, it is having both 5' to 3' as well as 3' to 5' exonuclease activity. So before knowing that, we have to see what are the activities present in a DNA polymerase. Like a DNA polymerase will always have a 5' to 3', 5' to 3' polymerase activity. What do you mean by polymerase activity? That means this DNA polymerase enzyme, it will add on the nucleotides in the DNA, in the daughter strand, in the 5' to 3' direction. Like suppose the DNA which is present, it is having two strands. That is, the one strand is 5' to 3' and the other strand is 3' to 5'. Okay, so the DNA replication starts from 5', from this side, 5' to 3'. Okay, and this is being done, this work of DNA replication is done by the polymerase activity of DNA polymerase and this DNA replication. So let us see what is the work of DNA polymerase 1. This DNA polymerase 1, it is having 5' to 3' and 3' to 5' exonuclease activity. So what do you mean by this? We have already discussed about the polymerase activity, but 5' to 3' exonuclease activity means when it goes forward, it eliminates the DNA. Okay, it eliminates or it removes the DNA in the 5' to 3' direction. What is 3' to 5'? Suppose this DNA is being replicated in this direction, that is from 5' to 3', and in this place there is some mismatch. So during this time, what happens? The DNA polymerase, it back turns and it removes one or two DNA which are mismatched. Again, the DNA polymerase activity will be carried out. DNA polymerase activity will be carried out, so that this DNA which was going on synthesizing the daughter strand, it will not show any error due to this 3' to 5' exonuclease activity. So the main work of this 3' to 5' exonuclease activity is that if it recognizes any mismatch, it backturns. This DNA polymerase will backturn, it will eliminate one nucleotide, then the DNA polymerase will again synthesize. Okay. So what is the function of DNA polymerase 2? This DNA polymerase 2 is having, the main function is it is having 3' to 5' exonuclease activity, which is mainly involved during this repair mechanism. And let us see, in this DNA polymerase 1, it is also responsible for the filling of gap after removal of RNA primer. Already we have discussed in the lagging strand there are different RNA primers which forms the Okazaki fragment. After this removal of this RNA, this DNA polymerase will synthesize a short strand, and it is done by 5 prime to 3 prime. First it will be removed, then it will be synthesized by the 5 prime to 3 prime polymerase, because it is a short stretch of DNA. Okay, so next is the DNA polymerase 3. This is having 3 prime to 5 prime exonuclease. It is not having 5 prime to 3 prime exonuclease activity. Only the 5 prime to 3 prime exonuclease activity was present in polymerase 1, which is not necessary. Okay, we can tell that it is not necessary that every time there will be 5 prime to 3 prime exonuclease activity. So why it is not necessary, why it is a waste process, let us discuss. Okay, like in this diagram we can see that here there is a DNA polymerase 1 and there is a nick present here. So for filling this nick, a DNA polymerase 1 can be used, because it is not having so much efficiency that it will synthesize a long stretch of DNA. That is the reason DNA polymerase 3 is a main enzyme for the process of DNA replication. So for synthesizing a short stretch of DNA polymerase 1 can be used. So this DNA polymerase 1, as it is having 5 prime to 3 prime exonuclease activity along with 5 prime to 3 prime polymerase activity, what happens? After synthesizing this, it will remove these nucleotides from here. Then it will again synthesize the strand. So there was not necessary that it has to remove this nucleotides. This is an energy. It is utilizing more energy and more time for this repair mechanism to take place. So this DNA polymerase 1 was divided into large and small fragment. One fragment was known as clinofragment. In this clinofragment, the 5 prime to 3 prime exonuclease activity was removed. So what happens in this clinofragment? We can see here that there is a gap present here, and by the use of DNA polymerase 1, the gap is filled. But the other nucleotides are not being removed. Existing nucleotides are not replaced when we use a clinofragment of DNA polymerase 1. Okay. So next is the DNA polymerase 2. This DNA polymerase 2 is mainly responsible for the DNA repair mechanism. It is having 5 prime to 3 prime polymerase activity. It is having 5 prime to 3 prime polymerase activity as well as 3 prime to 5 prime exonuclease activity. So what was the work of this exonuclease? When the DNA is synthesized, when the DNA or nucleotide is being synthesized, if it recognizes any mismatch, it backturns the DNA polymerase, it backturns and it removes this DNA. Then again the DNA polymerase 3 will do its work. Like the DNA polymerase, again it will synthesize the strand. This was the work of this DNA polymerases. Okay, already we have discussed about the 3 DNA polymerases, that is DNA polymerase 1, 2, 3. And the DNA polymerase 4 and 5, it is mainly responsible for the repair system. Okay, so this we will discuss when we will discuss about the repair mechanism. Now let us see how the structure of DNA polymerases. Here is the structure of DNA polymerases. It is having holo enzyme and core enzyme. Holo enzyme and core enzyme is present. The core enzyme, or we can tell that it is the catalytic subunit. It is the catalytic subunit. This alpha, theta and epsilon, this is known as the catalytic subunit. All put together, this is known as the holo enzyme. So what are the other things which are present apart from the catalytic subunit? This beta is known as the sliding clamp. Okay, it is known as a sliding clamp, and this is the clamp loader. This is the clamp loader, which is recognized as gamma complex. This whole total is this purple color one is known as the gamma complex, or it is also known as the clamp loader. Beta is known as the sliding clamp. Okay, so this tau is responsible for putting all together this subunit. 2 alpha subunit are being dimerized by the help of this tau protein. Okay, let us say very clear about this. Like we can see here, there is a clamp loader. This is a clamp loader which is composed of gamma subunit as well as tau subunit. This is the clamp which is known as beta. When this beta subunit is attached with this DNA, it requires ATP. So how this subunit is being attached with this DNA? At first, this DNA polymerase, this core enzyme which was present, which is known as alpha, epsilon and theta, it is a core subunit. 2 core subunits comes and binds. Okay, and this 2 core subunits, it is attached by this tau polymerase. Okay, it is the tau subunit. So here along with it there is also beta subunit as well as gamma subunit. This whole total is known as holoenzyme. It is known as holoenzyme. So let us now see how this polymerase is working. So during the process of DNA replication, already we have discussed in the basics of DNA replication that it is having a leading strand as well as lagging strand. So this DNA polymerases which are present, there are three DNA polymerases 3 working. One DNA polymerase is working in the leading strand, okay, and two DNA polymerases are working in the lagging strand. Okay, so in case of the lagging strand, there are multiple fragments, like there is a Okazaki fragment, so there is need of multiple DNA polymerases. Like when one subunit is working, before it is displaced, the other subunit can come and synthesizes the other part of the DNA fragment. Okay, so in this direction, in the leading strand, in one direction the DNA polymerase is going, it is synthesizing this and it is going in the forward direction. But in case of lagging strand, it is being synthesized in the opposite direction and it is done in a stretch. Suppose this polymerase is working here and this was the polymerases which were working. In the next, what happens, here another primer is being synthesized. So here in this time, if this DNA polymerase is not released, so something has to bind over here, so it is done by the other DNA polymerases. So that is requirement of the sliding clamp. So what is the requirement of the sliding clamp? This sliding clamp help in the processivity of DNA polymerase. The processivity is increased when the sliding clamp is present and it helps the DNA polymerases to maintain its place. So this is the main work of the sliding clamp. Main work is that it help in the processivity. Like we can see here that this is the 3 subunits. This DNA polymerase 3 is having 3 subunits. 3 core enzyme is present here. Here there is one. This is the third subunit. So this is a clamp loader. This C-like structure which we can see, this is the clamp loader. When it is open, it utilizes, during this time it opens, it utilizes ATP, and after it is opening, then only the DNA, it is being inserted into the DNA. So for loading the sliding clamp, it requires a loader. This is the sliding clamp loader. This is the sliding clamp loader. So this sliding clamp loader, for loading the sliding clamp into the DNA, it requires ATP. Okay, here there is 3 DNA polymerases, and for binding this, there is a linker. For this linking, it requires tau protein. Okay, so let us see, sliding clamp increases the processivity of DNA polymerase. Already we have discussed about this. What was the work of the sliding clamp? That is, it help in the processivity of DNA. So here we can see the sliding clamp loader which was present. It requires ATP. Like we can see here, this is a sliding clamp loader. It requires ATP. By utilizing this ATP, it transfer or it opens this sliding clamp. So this is a pentameric. It is a pentameric protein. It is a pentameric protein. It opens, we can see here this sliding clamp is open now and it is being attached and it is being loaded by the sliding clamp. This is the sliding clamp loader. Okay, and during this time there is also ATP hydrolysis, and this, after loading the sliding clamp loader is being removed. Okay, we have already discussed about this DNA polymerase 3. Like we can see here that how these are working. This is the DNA, and for the synthesis and for the synthesis of the DNA, both in the leading as well as lagging strand, the DNA polymerase works simultaneously. Okay, so here we can see that there was a thumb present. There was a thumb present as well as there was a finger were also present, and this was passing through the palm domain. Okay, let us see what are the other things which are present inside this thumb. Like when there is upcoming nucleotides, like here we are having cytosine, so the upcoming nucleotide is guanine for it, because adenine always base pairs with thymine by the help of 2 hydrogen bond, and guanine always base pairs with cytosine. So here we are having cytosine in the parent strand. So here the DNA, here the guanine have to come and bind over here. So when it is binding it, there are some other work of O helix of DNA polymerases. This DNA polymerases is having a O helix. Okay, so this O helix is having some of the amino acid like tyrosine, lysine and arginine. Okay, so the work of this tyrosine is that it will help by folding the fingers, by folding the thumb. We can tell that by folding the thumb to 40 degree, what is it gives a pressure to the guanine. Okay, it gives a pressure to the guanine to come and place here. Okay, and the lysine and arginine which is present, it interacts with this phosphate. It interacts with this phosphate. Which phosphate? Beta and gamma phosphate. And the 3' OH group which is present, it have a attack with this alpha phosphate. Okay, so this was a work of this O helix of DNA polymerases. So here we can see that when this thumb is pressed, here in this 3' OH group, the incoming nucleotide come and bind, which is complementary to the parent strand. Apart from this, how they will bring the new nucleotide, it is also having two domains like that. The one first is the polymerase domain, another one is the editing pocket. Okay, here we can see that this is the palm domain which was present. Here it is the polymerizing domain. Okay, this is the polymerizing domain where the polymerization takes place, or the upcoming nucleotide comes and bind in this region by passing this region. What happens if there will be any mismatch or there will be any error during the process of this polymerization? So during this time, this parent daughter strand which is present, it will twist itself towards this editing pocket. In this editing pocket, there will be exonucleases of the nucleotides which are mismatched. Then again it will turn near the polymerizing pocket and the polymerization will take place. So there are various type of DNTPs as well as RNTPs present inside the cell. So how a polymerase will decide that whether it is a DNTPs or RNTPs? So in case of DNTPs, there is 3' OH group is present, but in case of RNTPs, both 2' OH group is present as well as 3' OH group is present. So it has been recognized by a pocket. We can see here there is a pocket. If there is a correct nucleotide or if DNTPs is present, it will be fit in this region. Okay, but if other than this DNTPs, RNTPs is present, it cannot fit in this pocket. Okay, due to the steric hindrance, there will be no base pairings. In this way, it recognizes whether it is a DNTP or whether it is a RNTPs. Okay, here we can see that here there is a discriminator amino acid which is known as glutamine. This is known as glutamine, which is known as the discriminator amino acid. Okay, so here what happens, we can see here there is a DNTPs, only 3' OH group is present in this DNTP, so it is easy for it to get placed in this editing pocket. But if RNTPs is present, it is having 2 OH group in the 3' carbon as well as 2' carbon, it is having 2 OH group, so it cannot fit in this pocket. Okay, so now the question comes that if there is correct base pairing, then there will be a phosphodiester bond. But if there is incorrect base pairing, there will be no phosphodiester bond formed. So why it is so? So here we can see that it is a correct base pair. So what happens here, adenine is binding with thymine, so for this it needs a phosphodiester bond. So here if it is a correct base pair, it easily comes in this pocket, and the structure will be in such a way that this OH group which is present here, it will be closer to this alpha phosphate of this upcoming or the new nucleotide, so there will be a phosphodiester bond. But if there is an incorrect base pair, it cannot fit into this pocket, and the alpha phosphate of this upcoming nucleotide will be not nearer to this OH group. So there cannot be any attack or there cannot be any phosphodiester bond. Like we can see here that adenine is base pairing with thymine, so it is a correct base pair. So it does not require this. So it helps this nucleotide to come and bind in this pocket, and the distance between this alpha phosphate and this OH group is not far. But in this case, as this is adenine, it is also adenine. So here there cannot be any base pairing over here. So here the structure is in such a way that the phosphate group is present far apart. Okay, so apart from this discriminator amino acid, there are also other things which are used during this process of DNA replication, there are metal ion A and metal ion B. The metal ions, they are, this metal ion A is known as Mg2 plus and metal ion B is known as Zn2 plus. Okay, so this metal ion A, it binds with this OH group, hydrogen of OH group. Okay, but this metal ion B, which is zinc, it binds with this phosphate of the upcoming nucleotide. It helps in the stabilization of this things. Apart from this, there is also aspartate, okay, which help in this stabilization or linking of this two metal ions. This is also the same thing. Here there was a DNA polymerase. Inside this DNA polymerase, these all are the structures. Already we have also discussed these things. Like we have already discussed about the word that is it is having a processive enzyme. What happens if there is a processive enzyme? Or we can tell that what happens if the processivity is high and what happens if the processivity is low. Here we can see that here there is a non-processive DNA polymerase. Here we can see it is not processive. So here only one DNTP is added. After the adding of this one DNTP, this DNA polymerase is being removed. But if the processivity of the DNA polymerase is high, after adding many DNTPs, it will be removed. Okay, so here also we have discussed about this polymerization domain as well as the exonuclease active site or the editing pocket. Okay, this was all about this type of exonucleases, structure of exonucleases, the different type like DNA polymerase 1, DNA polymerase 2, DNA polymerase 2, DNA polymerase 3, their function, their structure. Already we have discussed in this video. In the next video, we will discuss about the prokaryotic as well as eukaryotic replication. In one video, we will discuss about the prokaryotic. In the other video, we will discuss about the eukaryotic DNA replication. Thank you for watching this video till the end. If you like this video, please do like, share and subscribe. Thank you.